not to cough into the microphone, so I'm cough. Okay, so on Friday we had our guest lecturer, which was your TA, Paul, and I'm just going to reiterate what he said about phage T4, and then we're going to segue into phage lambda. So the two differences are, one is a permanently lytic phage, and the other one is a lysogenic phage, which we'll go through today. So he went through phage T4, which is a lytic phage, and so here we have these two what we call coli phages, and the reason why they're called coli phages is because they are phages that are specific to E. coli, and that's how they got their name. And you can see that they look similar, but they also look different. So T4 is the one on the right, and that one's a little more complicated in its structure than phage lambda. And phage T4 is what we call a lytic virus or bacteria phage. Okay, so I just wanted to go over and remind you what he talked about in class. And so this particular phage is very interesting. So it can lyse a E. coli cell in 22 minutes, which is really fast in terms of how things go on a cellular basis. So it's this phage here, and it sits down and it docks down into receptors that it can bind to on the surface of this E. coli cell. And two things are happening, okay, when that happens. So the DNA is linear inside the capsid head. It's injected, and as soon as it's injected, it circularizes, okay? So there are two things happening. One is that this phage needs to replicate its own DNA. So this is what's going over here on this pathway. But it also needs to make some proteins that are specific to it. So that's what's happening over here. You can see transcription and translation happening. This is separate from DNA replication, okay? So to replicate, it circularizes and forms something similar to what we have seen before, right? It kind of looks like a plasmid, like a small circular piece of DNA. And it replicates through rolling circle replication. So it's just replicating around the circle many, many, many times. And that's a very efficient way of replicating. Some plasmids replicate like that as well, okay? And it forms this long chain, what we call concatemers. And a concatemer is just a genome linked to a genome, linked to a genome, linked to a genome. That's all coming off this rolling circle replication. So it just stretches and stretches of full-length genomes of that virus, okay? And that is important in the packaging, which I'll get to in just a moment. But at the same time, you also have this early messenger RNA synthesis going on, which is then translated by host ribosomes. And these proteins are involved in degrading the host chromosome. Okay, so they are nucleases. They degrade DNA, and so they're trying to get rid of the cellular DNA, get that out of the way so they can hijack all the polymerases and other machinery for itself. Okay, and then you have late genes that are transcribed and translated, and these are the ones that encode the capsid proteins and the tail fibers and everything that actually makes up the virus, right? So it's a timed process. If you want to have things to package, which in the things that you're packaging are your genome into these phage heads, okay? So this is happens after this. So you have a lot of these concatemers, and then you have all of your capsid proteins and tail fibers being created and assembled, and then you're packing the genome into the head of this phage particle, and then you're assembling the tail fiber, the tails and then the tail fibers, and then you have a viral encoded lysozyme, which is an enzyme that will degrade the cell wall of this E. coli cell, and the result is rupture of that cell and release of the virion particles. Okay, so that's that, and I just wanted to touch on this because it can get a little confusing for students. So this is the rolling circle replication that I was talking about. So this is the T4 chromosome that's circularized, and then it starts new DNA synthesis in a 5' to 3' fashion this way. Okay, so it's and then you have synthesis of the complementary DNA strand here, and then what happens is you have just chains and chains of those viral genomes linked back to back, okay? So it's important that a virus package all of the genes of its genome, right? So it has the capability to propagate itself in the future. It needs all of its own genes in order to do that. And so it really wants to make sure that its head is full of its own genome. And so T4 does this by packaging slightly more than it needs, okay? So there's a finite amount of space in this phage head. And so a head full of DNA is a specific amount of DNA. So how phage T4 ensures that it has all of its genes is that it packages its gene, it's a full-length genome's worth of DNA, plus like a little bit extra, okay? And so that means if you're looking at these genes and it's, for example, A to Z, right? Let's see, do I have on the next slide? Yeah. A to Z would be a full-length genome. It goes A and all the way to C. So it has A to Z, but it also has extra copies of A, B, and C. And it doesn't matter if you have extra copies, right? So then the next phage head will be full from D to F. And so you can see all the genes are represented, and you have like a few that are doubled, okay? And it just keeps going down the line like that. So there's always a full genome's worth of genes within that phage head plus like a couple extra. Okay, so that's how this one packages what we call a head full of DNA to ensure that it has all of its genes it needs. So if you were to look at the sequence of the genes in that linear DNA that's packaged into the phage head, they all begin and end at different points for phage T4, but they all have all the genes. Okay, so I'm going to just move into our today's lecture. I wanted to make sure that we solidify that concept of what a head full of DNA is and how that relates to T4 and how it's packaging with an offset. That's how it gets all of its genes taken care of or represented in the progeny. Way too many things here. Sorry about that. Okay, so now we're going to segue into phage lambda, also very exciting. Okay, so phage T4, its only choice, if you will, is to lyse a cell. So when it gets in there and it makes all of its genes and it packages everything, the end result is lysis of the E. coli cell and release of the virions out into the environment with the hopes of finding another poor, unsuspecting E. coli cell. Okay, so phage lambda has a bit of a more stealth lifestyle. Okay, so it can undergo the lytic cycle as well, but also what we call the lysogenic cycle. And this is termed a lysogenic bacteriophage because of that. And it's a more stable relationship. And so how a virus forms a lysogen is that it will integrate into the chromosome of the host cell, as you can see here. And when it integrates in, it's called either a lysogen or a prophage. And so those are two terms that are interchangeable. So once this virus has integrated into the host chromosome, it can be called a prophage or a lysogen. And if this process has happened, then lysogeny has occurred. Okay? All right. So the prophage DNA, and just let me clarify. So the prophage is the virus that's integrated in, but if you're a bacterium and you have a prophage, then you're called a lysogen. Okay? So this would be an E. coli lysogen because it has this prophage, a phage lambda, integrated into its genome. Okay, so it just gets replicated along with the host chromosome. So it's a quiescent relationship. It's not harmful to the cell. And it's beneficial to the virus because it's getting basically free replication without having to do anything. Now, it can enter the lytic cycle, and that's an induced process. Okay, so that's induced from what we know by DNA damage. So if a cell is undergoing a lot of DNA damage, the SOS response has been induced, and the virus can sense that the DNA is being degraded, then it will excise itself out of that genome and then start replicating itself and undergo the lytic cycle and lyse that E. coli cell and release its progeny out into the environment in order to save itself. Okay? So our example of this, there are many, many, many lysogenic phage, but our example we're going to talk about is the E. coli phage lambda. Okay, so this is a very, very well described and very well understood coli phage. And as you can see, it looks kind of like T4, but it's missing some of the fanciness at the bottom here. And it's a double-stranded DNA genome. It's also linear inside the phage head. Okay, so the genome is a linear double-stranded DNA molecule. And then it will circularize once injected into the host, very similar to T4. It is circularizing at these cohesive ends. Okay, so there's a single-stranded overhang here, and these recognize each other through homologous base pairing, and these are called the COS sites, or the cohesive end sites. Okay? And so these are 12 nucleotide complementary single-stranded stretches, and when it gets injected into the host, it circularizes via these COS sites. Okay? And the receptor on the E. coli surface is a porin. And it's actually the maltose porin, which is involved in bringing the sugar maltose into the cell. And so it's fairly well represented on the cell surface, and this phage lambda has evolved to recognize this protein here on the cell surface. And studies have even gone to figure out which residues or which amino acids in that protein are, in fact, the recognition sites that phage lambda is using. And so through point mutations and site-directed mutagenesis, researchers have found out that these up here, which makes sense, the ones that are exterior to the surface of the cell, are the ones that phage lambda bind to. Okay, so lysogenic phage are also called temperate phage. Okay, so that means they have like a good temper, if you want to think about it that way. And lytic phage are intemperate. So we're talking about a temperate phage here. And like I said, it injects its single-stranded chromosome into the cell, immediately circularizes via those COS sites, and then there's this sort of decision, if you will, to either undergo lysogeny, which is integrating itself into the chromosome, or to undergo the lytic cycle. Okay, and it can, backtrack on that decision, if you will, by if it decides to go this lysogenic pathway, it can reverse and go into this once this lytic cycle has been induced by DNA damage or something like that. Okay. All right. So again, definition of a prophage is a phage that is integrated into the host genome. Right. So this can happen pretty commonly, and it can also happen that this virus gets mutated and stays in the host chromosome forever. Like it mutates in such a way that it is no longer functional and it can't excise itself back out. And so if you look, if you're doing computational comparative genomics of bacteria, there are a lot of what we call phage remnants in the host chromosome. We don't exactly know what role those play quite yet in the physiology of the bacteria, of a specific species of bacteria, but we do know that there are phage remnants that are being dragged along in the chromosome as the cell is replicating. Okay? So prophage integrates in, and that results in lysogeny, and the bacterium that it has this is harboring this prophage is called a lysogen. Okay, so again, it has this decision to either undergo the lytic cycle or enter the lysogenic cycle. And this is just an outline of that. Okay, so the lytic cycle is very similar to phage T4. It gets in there, circularizes, starts replicating itself, and then packaging itself, and then lyses the cell and releases itself. Or you have lysogeny, where it integrates in and is still replicated as the host is replicating its own DNA, and it segregates into the daughter cells just as the chromosome would. Okay, so we're going to watch a little animation and then a very cool YouTube video that lasts literally like 8 seconds. And so I'll play it a couple times, but it's cool. So there was a lab that was able to take video lapse movies of E. coli cells that were lysing or had entered the lysogenic phase. Oh, and this is the paper I was reading that has the movie. So they were trying to figure out whether or not the decision of the lytic cycle versus the lysogenic cycle is random, or if there's some biological factor. And they showed that cell size actually plays a role in whether or not an E. coli cell will become a lysogen, or if the phage will undergo the lytic cycle.
Unlike a virulent phage, a temperate phage, such as phage lambda, does not necessarily cause lysis after entering a host cell. Instead, the temperate phage may embark on a pathway called lysogeny. In lysogeny, the phage first attaches to its host and injects its genetic material inside the bacterial cell. Linear double-stranded DNA is then circularized. The circularized genome then recombines into that of the host by site-specific recombination of DNA. The integrated phage genome is called a prophage. Lysogeny is a condition in which the phage genome is replicated along with that of the host cell as the host reproduces. Implicit in the term lysogeny is the ability of such a strain to generate a lytic burst of phage. The regulatory switch between lysis and lysogeny responds to environmental cues. That is, how likely is it that the host cell will survive and continue to propagate the phage genome under present environmental conditions? If the cell's growth is strong, it is more likely that the phage DNA will remain inactive, whereas events that threaten host survival will trigger a lytic burst. At the beginning of the lytic pathway, the phage genome directs its own excision from the host genome. As it exits, it circularizes again. A viral DNase cleaves the host cell's DNA. Using information in the viral genome, the cell synthesizes capsid proteins. The cell replicates the lambda phage DNA by a rolling circle method, making many copies that become packaged into the capsids. A phage enzyme then lyses the cell, and the progeny phage spill out, ready to infect another host cell.
Okay, so that's misogyny. Okay, so this is 8 hours of filming E. coli under a microscope, sped up into about 8 seconds. Okay, so I'll just let you guys watch it, and then I'm going to replay it a couple times. Big screen. Okay, now I'm going to pause it, and I'm going to explain what you're going to see. Okay, so you'll see up here, you'll see down here and up here that these cells start to fluoresce. So they've been labeled, or they've been infected with a phage that can express GFP, okay, phage lambda. And down here you'll see that this becomes a lysogen, so like a stable situation. So these start to fluoresce, and then they start to divide, and the daughter cells are also fluorescing. So that phage lambda has integrated into that chromosome and continues to express GFP as that process goes through the replication and binary fission cycle. Up here you'll see that these bacteria get infected, they start glowing, indicating that infection has occurred, but then they burst. Okay? So this is lytic versus lysogenic. It's pretty cool if they can capture that. For fun. And then the ones that aren't glowing are ones that did not get infected. Okay, so that's that. Okay.
Are you trying to learn Adobe Illustrator for designing beautiful graphics, logos, and more? My name is Phil Ebener, and I've helped thousands of people just...
Whoever that guy is. I don't like YouTube, how it just does that. Continual spiral. Okay. So like I mentioned, and like animation mentioned, there are some environmental cues that will trigger the Lytic cycle, and so you can stimulate this or induce this by DNA damage that is incurred by exposure to UV light or to chemical mutagens, or if the SOS response has been activated. And if we can recall, the SOS response is when there has been a lot of DNA damage and the cell is trying to just replicate its DNA as quickly as possible using very sloppy polymerases, but it's better at that time to replicate your DNA and not worry about error rate and things like that, because your DNA is being degraded. And so in this case, the phage lambda will excise itself out of the bacterial genome, and this happens through transcription of this excise gene or XIS gene, which encodes a protein or an enzyme that is involved in excision of the DNA from the host, okay? And just sort of a real-world connection that you don't need to know for the test, but in case you are doing cloning in the lab. So we talked about phage attachment sites, those ATT sites, and how that's where phage will integrate into the host chromosome sites. And then we also just talked about these excise proteins and excision sites. And so biotech companies have capitalized on this to use this for molecular cloning. So we talked about how we make recombinant DNA molecules and how we do that by cutting and pasting with restriction enzymes. You can also do it by cutting and pasting with phage proteins. So using the ATT sites and also the excision sites and the proteins that stimulate the integration and the cutting out. So that's just another way you can do cloning, and it's called, blanking, gateway cloning, sorry. Okay, so phage lambda again, right? So it circularizes in the host cell via the COS sites, and then how it gets packaged is it's cut right at the COS sites, okay? So there are these recognition sites that are inherent to those repeats on the ends of the chromosome, and that's where the phage will cut and package this into there. So it's a little more precise than T4, which just takes a genome's worth plus a little extra. This one recognizes specific sites, which is the COS sites, okay? And so each phage lambda has a full genome worth with those little single-stranded COS sites at the end. Okay, so replication of the lambda genome is through two ways, right? Okay, so it's it circularizes right when you get into the host, and then you have bidirectional replication. This is exactly like how the chromosome replicates. It starts replicating bidirectionally, okay? And that's just a little bit. That's right at the beginning, okay? That's just to give a few copies more than just one copy. And then to really get things rolling, it starts with the rolling circle replication. So all these circles that originate from this circle-to-circle or bidirectional replication start replicating through rolling circle replication. And that's where you can get really quickly a lot of copies of the genome in a short amount of time. Right? So you have, again, concatenamers, and that's just genomes linked to genomes in a long linear form. So this would be, for example, one lambda genome, and here would be another, and then more and more are rolling off the pipeline through rolling circle replication. It cuts at the COS site and packages that. Okay, so just to compare, and perhaps this could be an exam question, of comparing and contrasting lambda and T4. So they're similar, but they're different, right? So they're both coli phages that infect E. coli, but they have some differences to them. And so T4 is always lytic, like Paul taught you, And phage lambda, like I just taught you, is usually lysogenic, but it can be lytic when induced. And so we saw that in our video. You can see the differences between the lytic cycle and the lysogenic cycle. The DNA for T4, so they're both linear inside the capsid head. And when injected, they both circularize. Here they circularize with terminal repeats for T4, sort of through like a little mini homologous recombination. And then here they circularize via those single-stranded cohesive ends, or the cos sites. In T4, you have rolling circle replication, so it just immediately starts making lots and lots of concatemers via rolling circle replication. Here you have a little bit of bidirectional replication, and in those circles start replicating through rolling circle replication for phage lambda. Here for phage T4, like I told you, it cuts according to the DNA, plus a little bit extra, so about 103% of the genome size. And for phage lambda, it cuts a specific cos site. Okay, and just to loop this back to what we've been learning in terms of biology of bacteria and their relationship with phage, so we talked about transduction. So transduction is moving host genes around using a virus, okay? And we talked about generalized and specialized. So just to remind you, generalized is where some DNA of the host accidentally gets packaged into these phage heads and released into the environment. This new phage then comes and injects this DNA into a host, and through homologous recombination, it will recombine into the host chromosome and become a part of that. And through natural selection, if it's something beneficial, it will maintain that gene or genes in that particular species of bacteria. And this is lytic, okay? So this is where generalized can be any piece of DNA from the host chromosome, because you can see here that part of this lytic cycle is immediate degradation of the host chromosome. So you have lots of bits and pieces of the host DNA floating around, and you can imagine it's very easy to get some of that accidentally packaged into these phage heads. Okay, so any DNA is susceptible to this in the host. Whereas specialized, only DNA that is adjacent to where the prophage integrated can be transferred. Okay, so this is where you have a lysogenic phage that integrates into the prophage. Induction of the lytic cycle occurs. It excises to those excise sites in the XIS proteins that I just told you about. And sometimes there is incorrect or less precise excision. And so it takes up a little bit of the host DNA that's adjacent to those phage attachment or ATP sites here. Okay? And so specialized just means only the genes adjacent to the phage the prophage can be transferred. Okay, so that is bacteriophage in a nutshell. So we only talked about two, and we talked about two examples of lytic and lysogenic. But I want you to recognize that there are many, 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 many prophage out there, many, many, many bacteriophage out there. Okay, so basically, I've been told by viral ecologists that if there's a bacteria, there is a phage that will infect it. So there are many, many, many types of these viruses that are very important for the ecology of microbes in the environment, and we're just now beginning to learn what role they play in terms of cycling of nutrients, because you can imagine that blowing up a bacterial cell releases a lot of nutrients back out into the environment, and also through gene exchange, and also through natural selection of bringing beneficial genes into specific species of bacteria. They're generally very specific to a bacterial species. So, for example, it's very hard to get a phage that would infect E. coli to affect something like Xanthomonas. Okay, so they're not very interchangeable, and you're very lucky if you have a bacterial system that you can create a transducing phage for. So you can have a phage that will move DNA for you. I am not so lucky to have that for my system. If you work in E. coli, you can use these. Okay, so now we're going to move on to another one of my favorite topics of microbiology, and that is microbial pathogenesis. So I am a pathologist. Oops, sorry. I need to close my email. It's like my greatest fear is that I'm going to get some email during class or some Skype call. It's some message. microbial pathogenesis. Again, I am a pathologist. I study plant pathology, so I study bacteria in particular that infect plants and cause disease. But obviously, you know that there are microbes that are pathogenic on almost every organism on Earth, or if not every organism on Earth. Every organism has a pathogen, okay? So that can be anything, ranging from humans to animals to fish, to plants like I just mentioned, to birds, reptiles, cows, everything. And even bacteria, right? So we just talked about bacteriophage. Those can be considered pathogens of bacteria. And so in all of this, we try to understand how these bacteria are causing disease, and if so, can we utilize any of that information to control disease? Okay, so generally, as pathologists, we're looking for what we call virulence factors. And we'll talk about virulence versus pathogenicity in just a second. But a virulence factor is something that enhances the disease-causing abilities of an organism. Okay, so if you are creating, say, a random transposon mutant bank and you're screening it for virulence, you would put all of those mutants into your host of choice. My host of choice is plants. And so I then screen all of my plants and I look for the ones that don't get us sick. And then I go and sequence that transposon and I find what gene it's in, and then I say, oh look, I've discovered a new virulence factor for this bacteria, because when I remove it from the system, the bacteria is not as good at causing disease. And perhaps that's a target for therapeutics. Okay? Yes? She's asking what's wrong with the mouse. I'm not sure. There's any variety of organisms that can attack a mouse, and that one actually doesn't look very sick. Well, we can pretend it has like cholera or something. I don't know. I borrowed this slide from Dr. Long, who also teaches this class. So I don't know what that is. Okay, so there are different types of symbioses that we're going to be talking about for the rest of the quarter. And symbiosis just means that two organisms are living together in an intimate relationship, okay? That relationship can be great for both of them. It can be good for one and sort of for the other, or it can be bad for one where the other one is benefiting, okay? So commensalism is where one benefits and the other one is seemingly non-affected, okay? So, for example, it's thought that many of your gut microbes are commensals in your body because you are benefiting from them. You are letting them digest your food, you're letting them enhance your immune system, and they're in there and they're seemingly not really effective, okay? Mutualism, where both organisms benefit. We'll talk about that when we talk about nitrogen-fixing bacteria. But today we're going to talk about this, which is parasitism, where one organism is benefiting, which is the microbe, and the other one is harmed, which is the host, which is, in this case, we're going to be talking about eukaryotic hosts, so either humans, animals, or plants. Okay. Moving on. So a couple definitions we have to go through as we start to embark on this pathology journey. So infection just means that a bacteria or a fungus or a virus has entered and can multiply within another organism, right? Infection doesn't always result in disease, right? So you can get infected with something and you can defeat it with your immune system. So for example, this cold or whatever I have, if I was able to defeat it, it would have gotten inside of me and started multiplying, but my immune system would have removed it, which obviously that was not the case this time. But it doesn't always cause disease. And so my graduate student and I are actually writing a paper now, and we keep falling back onto different stages of infection. I'm like, no, it's not stages of infection that we're studying, it's stages of disease that we're studying. So we have to always make sure that we're mindful that we're using the right terminology. So infection just means that the organism gets into another organism, multiplies, but it doesn't always mean that disease is the result. Okay? Now, infectious disease is when this multiplication of the pathogen results in disease. Okay? And that just, what is disease? Okay, that just means that some or parts of the host functions are not functioning properly. Okay? For example, my throat is not functioning properly today. Okay, so we're going to focus really on bacterial infections because we just have to focus on something. And there are these different steps. And generally, they all fall into this. So you have to attach to your host. Attachment is very important. So remember biofilms, attachment is the first step in that, and it's very important for bacteria to propagate themselves in an environment. That's why they're so closely linked to pathology, because attachment is one of the first steps of infecting a host. And then they have to invade. They have to get inside the host tissue. Okay? They have to somehow circumvent or suppress the host defense responses. So if that's a plant which only has innate immunity, it somehow has to suppress that. Or if that is a human, you also have to suppress innate immunity, but also circumvent adaptive immunity. And then you have to acquire nutrients from the host so you can survive and live in the host. Then you have to grow or propagate in the host and multiply yourself. And then, ultimately, you need to transmit yourself to a new host. So these are the steps of bacterial infections. And there, it runs the gamut of many different choices here. But to continue on our definition journey, so when we're talking about host-pathogen interactions, we talk about pathogenesis. And that's basically the process by which a microbe can cause disease in a host. So a pathogen, if you can cause disease, you are considered pathogenic. Okay? A parasite versus a pathogen. So the two terms are really sort of interchangeable, but depending on what literature you're reading, you will see that pathogens generally are talking about bacterial, fungal, and viral pathogens. When you're talking about parasites, eukaryotes, like protozoa and stuff start to come into play, but really parasite and pathogen are the same thing. There's something that, benefiting from another host and while harming the host. Okay? And they can enter and infect their host in different ways. So they have a variety of strategies. Okay, and so we talked about infection and what that means, and that's just the entryway or how a particular pathogen or parasite gets in. So if you're a respiratory virus, you generally enter through the respiratory system, right? If you are a wound pathogen, you enter through a wound. If you are a stomach or a gut pathogen, like E. coli, you enter in through the alimentary canal or the food canal. Makes sense, right? If you're a bloodborne pathogen, you enter through a wound that enters you straight into the bloodstream. So there are different entryways to get in. And again, infection doesn't always mean disease, because a lot of infections are removed by the immune system. So we're constantly assaulted with different pathogens in our environment on a daily basis. We're not sick every day, hopefully, and that's because our immune system is really good at clearing out these bad guys. Okay, so in terms of pathogens, we have primary pathogens and opportunistic ones. So a primary pathogen means that it is a pathogen in its own right. Okay, so it can enter you, it can cause disease, and it has the ability to sort of overcome your host defense system. There are also opportunistic pathogens that might be residing in you as seemingly a commensal or something that's not harming you, but then when something is sort of breaking down in the system, they take the opportunity to then become pathogenic. So dysbiosis happens in your microbiota, and a good example of this is like Helicobacter pylori, which lives in your stomach. It's the causal agent of stomach ulcers, and it's there, it's in your microbiome. But then when something happens where you become stressed or your immune system is now out of whack, it can flourish and bloom in your stomach and cause ulcers. Okay, so that's an opportunistic pathogen. It could also be that, you have staphylococcus on your skin, it's living there, it's not causing you any problem until you cut yourself, you don't clean it properly, and then it infects and you get a staph infection. So that's an opportunistic infection. Okay, there's also a latent state, so the most good example of this is the herpes virus. So if you've ever had cold sores, most of the time they're in a latent state, this virus, right? Until you become stressed or compromised in some way, if your finals weak or something, and then your cold sore appears. And so you know that if you have cold sores, then you're harboring this herpes virus in your epithelial cells, and then it will sort of bloom out under these times of stress. So they can remain dormant for years and then suddenly emerge depending on the situation. All right, so what is pathogenicity versus virulence? Yeah, that's the next slide. Okay, so a pathogen is the ability of an organism to cause disease. You're either a pathogen or you are not a pathogen, okay? Infectivity is how easily this can cause disease. And virulence is a measure of how severe the disease is, okay? So virulence and pathogenicity are another two of those terms that people like to use interchangeably, but there's some nuances to them, okay? So virulence just means how severe of a pathogen you are. So either you are a pathogen or not, so that's a plus or minus situation, a yes or no. But depending on how virulent you are is a spectrum, okay? So you can be have low virulence and still be a pathogen. So for example, like the rhinovirus, which causes the common cold, it's highly infective but has fairly low virulence, right? Like a lot of people don't die from the common cold. You get it, and then there's low virulence. Now, Ebola is highly virulent because a lot of times, 70% of the times, it can be fatal. And so virulence is just a measure of the degree of how much of a pathogen you are. Okay, so I just want to reiterate that. This is something that I ask all graduate students in the plant pathology program if I'm on a qualifying exam. Please define these two things. And again, pathogenicity is a plus or minus situation. So this is the organism can cause disease or not. Virulence is a measure of the degree or severity of disease. So, for example, going back to our example with the transposon mutant bank, and we're screening for what we call virulence factors. In that case, we're looking at factors where there's less disease in this situation. So we have our wild type, which causes disease quite well, and then you can see that you've knocked down virulence in sets of your mutants. Okay, so you can say, oh, we have a low virulent phenotype over here, medium virulence, and here's our wild type virulence. And so it's an additive effect. And so somehow you've knocked out something that is required, but not 100% required for pathogenicity, but you've decreased virulence. Now, if you hit on something where you have no disease, then you get really excited because you say, oh, I found a pathogenicity factor, not just a virulence factor, but a pathogenicity factor. And that means it's absolutely required for that organism to cause disease. And so that makes for a really nice title of the paper, if you find that. Okay, so we're going to leave off there for today.